Good morning, my name is Catherine Bonilla, and today I'm going to be presenting my second evaluation of molecular biology techniques involved in the creation of peptide-based COVID vaccines. As a result of the global pandemic, experts from all over the world set out to make COVID-19 vaccines that were both safe and successful. Molecular biology methods were used to make a peptide-based vaccine, which was a very interesting idea. In this video, we will be looking into the complicated science that made this possible. To begin with, you need to understand the virus. COVID-19 was caused by the virus SARS-CoV-2, which has a unique structure made up of different proteins, including a spike protein on its surface. This spike protein is very important for viruses to get into human cells and is a great target for making vaccines. Researchers found out that some areas of the spike protein, referred to as epitopes, when recognized by the immune system, cause a strong reaction. These epitopes are essential for creating a vaccine that works and produces protective immunity. Now, let's examine the molecular biology techniques applied in the development of the peptide-based COVID vaccine. To begin with, computer programs were utilized to examine the genetic coding of the virus and identify the spike protein epitopes that elicit the most immune reactions. The choice of peptide patterns for the vaccine was greatly aided by the bioinformatics studies. Synthetic biology techniques as well were employed to create the peptide sequences associated with the identified target epitopes in the laboratory. These peptides in the vaccination represent antigens. With peptides in hand, the next step was to formulate them into a vaccine capable of simulating a robust immune response. To improve the peptide stability and immunogenicity, scientists combined them with adjuvants and other ingredients. Adjuvants are compounds that strengthen the immune system's reaction to the vaccination, guaranteeing a more robust and durable protection. Before testing in humans, the peptide-based vaccination is preclinically evaluated in animal models, usually mice. In these investigations, the peptide-based vaccine is administered to mice, and the ELISA and flow cytometry methods are used to assess the mice's immunological response. Epitope mapping is then conducted to determine which region of the S protein and targeted by antibodies generated by the vaccine. This involves testing the antibodies against a panel of overlapping peptides covering the entire S protein. Expert immunological tests were utilized to evaluate the vaccine's capacity to elicit humoral and cellular immune reactions, two important components of the immune system's defense against viral infections. The generation of antibodies and T-cell activation were assessed during these assays. Using particular peptide sequences obtained from the spike protein of SARS-CoV-2 and combined with adjuvants to increase immunogenicity, the peptide-based COVID vaccine provides a tailored method to elicit a strong immune response against COVID-19. The successful use of these molecular biology approaches led to the creation of a COVID vaccination that is peptide-based and presents a viable means of containing the current epidemic. Worldwide immunization initiatives are ongoing, and because to scientists unceasing work, there is hope for a healthier and better future. Together, we are stronger than ever in our battle against COVID-19, and thanks to the power of molecular biology and the various vaccines that were created, we could get to control the various diseases that attack our immune system and our world every single day. Thank you very much, and I hope you enjoyed.